Dr. Jishy. Thanks for accepting our offer and sitting with us to talk about your work and life.
Sure.
Let's start with your journey to becoming a scientist. How did your school, college experiences shape your decision into becoming a researcher?
I grew up in an environment where books were like really a big deal. So my father used to get books at home, and this was really influential in my early childhood. So he actually got a lot of science books as well. Right. So at the time, I think I'm talking about early 80s, and a lot of USSR was still existing, and we used to get a lot of Russian books translated in English, which were available in India. So we had a lot of Russian science books available at home, with a lot of models about how rockets are launched, and how space shuttles work and stuff like that. So this was like very early on. I had those books, and then there were also books which were much more serious. Like we had books on I don't know this was in Malayalam, so I don't remember the exact translation. Right. I still have the book here. Right. So it's a book from 80s, written by Yakub Perelmo. So he wrote about a lot of scientific experiments which happened, early science experiments where people tried to defy gravity, people tried to make machines which were like, which would never stop and which would function on its own. It doesn't require any external help, but they would function on its own. And H.G. Wells's stories and stuff like that. So a lot of the information was available in this book. So, and then I also had a bit of academic background, which was helpful in the family because my grandfather did a BA honors in physics. So at the time he went to St. Joseph's College in Trichy, and he studied there, which was a rare thing. And my grandfather's brother actually did a PhD in the Yale University in 1930s. So very, very early on, there was some academic environment in the family. Again, I cannot claim that influenced me in one way or the other. But I think towards the end of my schooling, I kind of realized that I like science more than other subjects, but I was not sure which way to go. Like I liked physics, chemistry and biology to the same level. And then during my later schooling, I realized that maybe biology is better. So I got more interested in biology at that point of time. But again, I was a child who was not spending a lot of time in a structured learning this thing. I was not studying much at all. My parents were really worried. So I would listen to the class and then that's it. I would come home and would be in my own world. I explored a lot of my surroundings, and I was really inquisitive over things. Like for example, I remember very early experiments which were done with ants, and to figure out like the pheromones which like they kind of realized that they are traveling through a particular track. If you break the track, what would happen? If you remove the pheromones from there, I didn't know that it was pheromones. I knew that there is something secreted by them. I would also try to remove the pheromone from one place and try to apply it to other place to see whether they will get attracted to that or not. So a lot of experiments with ants, a lot of experiments with plants, like looking at budding and the way in which plants grow in different directions. And I think this was the real nurturing of the scientists in me. I think that's where it started. But the real scientist in me came out during my PhD time at NCBS. I think this was a real training ground for the scientists in me. I think I had a great mentor, Dr. Apurva Sareen. She gave me a lot of freedom. I was the first student in the lab. So that again gave me a lot of freedom to explore things. So I think the real scientific growth happened from NCBS, which continued in my postdoc mentor's lab, Steve Cohen's lab. Right. Again, he was a great guy, like, which gave me a lot of freedom. I came from a mammalian field and moved to the drosophila area, but he gave me enough freedom to explore things and then finally decide on a project and stuff like that. So that's my scientific growth. I think that's how I'm here. I don't think I can claim that very early on I had any special interest in science, but it happened like gradually on the way. It was a long path, I would say. I was not a very competitive student in school in early time. I was just about the top 10 in the class or so. Never the best student.
That is still the top.
Right. So I don't know. Yeah. That's my scientific journey. That's the journey which has taken me here. Right. Yeah.
So after your PhD, you moved on to do a postdoc, and you finally ended up in IISER Thiruvananthapuram.
Yeah.
And IISER was sort of getting established at that point of time. And being a young PI, at that time would have been challenging.
Yeah.
So how did you end up in IISER, and what were some of the challenges that you faced back then?
That's a very interesting question, because I only went to two interviews actually before I tried IISER TVM. Those were abroad, and that didn't work out. But then at the right time I came to IISER Pune. I met Dr. Shashi there, and we had a lengthy conversation about coming back to India. And he really told me that IISER TVM is there and it's a great place. It's a very young institute. There are very few people in biology at that point of time, and it's probably a right place for you to look at. And I came here, met people, gave a talk, and then I really liked it. So we were in the transit campus in the CET, College of Engineering, Trivandrum, in the city. We had like minimal facilities there, but whatever I saw there was very impressive, and I really liked it. I found that people were like very close-knit, were very friendly. There was a lot of interactions between groups. People were sharing labs with each other. So 3 labs would be sharing a small space. So that was a very cordial, very friendly atmosphere where people had to share things. And that clicked in me. So that was the main decision-making point was that. And then people convinced me to join here, like, people whom I spoke to, like really spoke great about this place. And already I could see the productivity, like there were good publications coming out of this institute, and people were establishing themselves really well, even with the limited conditions they had. And then the Vitra campus, I also came to visit this place, and I realized that it's a very scenic, beautiful place with a lot of restrictions, because one cannot access the city very easily. But nevertheless, given all that, I still prefer to be in such an environment, away from the clutters of the city and disturbance of the city. So I think these were really the decision-making points which led me to IISER TVM, right? And,
so how was setting up a lab in the initial days? Like,
setting up was really tough, because when I joined there was no fly room. So this was again a thing which attracted me to IISER TVM, not having the fly room, but to be the first fly person to be here. So which means that you have to start everything from the scratch. And I already had good experience with setting up things. I told you I was Apurva's first student. So that way I helped her in setting up the lab there. Then I moved to Steve Kahn's lab, and he moved from Germany to Singapore. Right? Again, there was a lab move which was involved there. And I helped the lab a lot in establishing in a transcontinental fashion, establishing a lab in a new place. And then within Singapore, the lab again moved to another institute. So I have experience of establishing like 3 different labs, which helped me a lot in starting up here. So I knew each and every detail of what I need here. I knew already before I came. So this helped me a lot. And then we had the support. There was good support from the institute for starting things. So the fly room had to be built physically. There was no place for the fly room, so we made a room, which took some time. But except that, I think everything went very smoothly. It was challenging. Getting flies here was also challenging. We had to bring about 300 to 400 lines to India from Singapore and from Germany, which was really challenging. But they all came and they were alive. It was, I think, being planned well helped. The administration was also very supportive at that time. Even now it's very supportive, but at that time it was a beginning place, so there was special help and special care given to people. I think that I enjoyed, and it helped me to start the place really well at that time. Right. So that's it's not easy, because you are coming back to India. There are challenges involved there for sure, which is different from when you are establishing a group abroad in a developed country. So that is always there. But this experience is also unique, and I'm sure you will get to the point like very easily. It's not a huge problem.
Yeah, it's a very interesting journey. Let's get into your area of research. What are the questions that you are interested in answering, and what are the projects going on in your lab?
In the lab, right? Yeah. So the fundamental, the basic, very overarching question which we address in the lab is how does the environment influence an organism? Right? Its development, its function, and its ability to survive during changes which can happen in the environment. So we basically look at, our major interest is to look at changes in the environment in terms of changes in the levels of nutrients in the environment and how that can affect an organism. So an organism which is living in the wild can actually experience that on a daily basis, or like an hourly basis even, or like a seasonal basis and stuff like that. So how do they survive with these fluctuations and maintain a normal equilibrium? Internal equilibrium is what we are interested in. So then it comes to the question, like, how does the organism sense the change in nutrient levels, and how does it elicit the various responses which are required to sustain during the changes in the nutrient levels. So we basically do that. We study ways in which the organism can sense the changes in the environment, and then also how does these changes would affect growth of the organism, development of the organism, and then the adult physiology of the organism. These are the basic questions we address in the lab. So basically, we work with Drosophila, and Drosophila has this main two tissues which are really important in sensing nutrient levels. One is the fat body, which is equivalent to the liver and adipocytes in higher organisms. The second is a set of neurons which is present in the brain, which produces insulin-like peptides equivalent to mammalian insulin in the fly. So these are two different tissues which are really important for sensing the changes in nutrient levels and managing the responses to changes in nutrient levels. Right? So our main focus are these two tissues, and then we have various projects which we are working on. So one project is to look at the function of this small non-coding RNAs called microRNAs, and how does they regulate gene expression and help to maintain the homeostasis, like maintain the internal balance, is one question which we are addressing. So that again is like, we have several sub-projects on that. Jarvis is my PhD student. He looks at the regulation of circadian rhythm, and he looks at microRNAs which are acting in the brain. It controls the circadian rhythm, sleep-wake cycle. He also has a project in which he was working on the aging process and neurodegeneration as well. So Jarvis, you can maybe talk a bit, very short sentences about what you're doing.
Yeah, so
you can take your mask off if you want, if you don't mind. Yeah, okay. Sure.
I work on microRNAs and their role on various aspects in regulation of basically circadian rhythm, lifespan and aging, and as well as growth and development. And basically, we're trying to identify microRNAs that perform, regulate such functions, and ultimately identify the targets that the microRNA regulates, and possibly look at the normal roles that these microRNAs could play that are conserved across the evolutionary tree right up to humans.
Thanks.
So Jarvis is a PhD student, and he would be like, he has written his thesis, and his thesis exam should happen very soon, I think. Yeah, sure. Good. Thanks, Jarvis. We are also interested in the effects of changes which are happening at different life stages, right? Okay. So how does a juvenile life stage change can affect the adult organism, or how does a change which can occur during the early adulthood can affect the later stages of life or later adulthood. So we are particularly interested in looking at the nutrient stress during these early life stages, and how would it affect the metabolism and the physiology of the organism at a later stage. So this has a lot of relevance again to human health, because juvenile stages, we know about situations where young babies can be affected by malnutrition or imbalances in nutrient levels, imbalances in oxygen levels, and so on and so forth. How would that affect the adult stages, or a grown-up human being physiology? How can they get affected? We are kind of creating a model to understand that, right, using the Drosophila system. So that is again like a very important project which is going on in the lab. We are also looking at the link between the nutrient absorbing, the nutrient sensing, and the nutrient balance system to the respiratory system of the organism as well. So oxygen plays a very important role in metabolizing things. It plays a very important role as being one of the major building blocks in the growth of the tissues and growth of the cells and stuff like that. So oxygen plays a very important role, right? So we are finding new connections between the oxygen sensing mechanism as well as the nutrient sensing mechanism, by which we now know that there's a crosstalk between these two. And attaining the final size of the organism, attaining proper development, requires a proper crosstalk between these systems. Again, this has a lot of relevance, because you can have like kids who grow up with chronic lung diseases, lung disorders, and then we will try to understand, we'll try to model a mechanism by looking at how this can affect the adult functioning, right? Adult physiology and adult functioning. So these are some of the projects which are going on in the lab, and we have a lot of other projects as well, but it takes a lot of time to explain that. But in a nutshell, that's what we do. So we look at changes in the nutrient environment and how does it affect the normal functioning, normal development, and normal growth of the organism. That's in a nutshell what we do in the lab, right? So this is the main area, one of the main areas of work in the lab. There's a molecular biology area where we do quantitative PCRs to look at gene expression changes. We have a Western blotting system to look at protein changes in levels of protein. So that's where all the molecular work happens. Okay. So we have a separate area where we do the fly work, drosophila research, and we can go there, right? Cool. Travis. Yes, we are. Hi.
So this is the sitting
This is where the students sit.
That's
an area where they can sit and read and or relax also.
Right.
Yeah. Cool. Please go. Hi. This is the fly room where we do the fly work.
Right.
So basically the handling of the flies happen here. They are living organisms, right? There is no way to freeze them or store them away just like that. So you have to keep them alive and keep them fed and keep them happy. So that's very important. So they are kept usually in the incubator room. There's a separate room. We'll go there. But then we bring them here and we take care of them regularly, right? Then they're also taken out to set up certain crosses by which we can express genes of our interest in different regions of the body. So there are systems by which you can express genes of interest or downregulate the expression of gene of interest in specific regions of the body. Okay, so there are tools which are available in the system. And by doing that, then you can study various phenotypes, right? What would be the outcome on growth, what would be the outcome on development and physiology and so on and so forth. So that happens here. So these are the regular microscopes which we use in the lab. This is a regular stereo microscope. And we have a carbon dioxide anesthetic system. So these are the cylinders where we have the CO2 stored, and then they get supplied to each station. And the CO2 would come here, and we use CO2 to anesthetize the fly, right? So you can't handle them when they are like moving around, right? So you anesthetize them and you put them on the pad. They will remain asleep for some time, right? So this will affect the nervous system actually. So it kind of slightly affects them. So you can't do an experiment right away after doing that. So you have to wait for a certain period for them to recover completely. But handling them, crossing them, looking at their phenotypes and stuff like that can be done in this setting, under this setting. Okay, so you can look at flies through the microscope, and then using a brush you can handle them. That's what we normally do. Okay.
These are
here is a vial of Drosophila, so you can see different stages of the Drosophila there. So you can see the larval stages, which are mostly in the food. You can also see some pupae here. They are like they pupate and then they stick to the wall of the tube. And then adult flies are also there. I think there are not too many adult flies here, but they are in the tube. So maybe we can look at another tube which has adult flies in them. So they are very small. It has to be handled under the microscope. So that's the thing. And this is specialized medium which we provide, which mostly has yeast, dextrose, and corn. Okay. And it's made by mixing agar and melting it, so it solidifies in the end, and it's easy for the larvae to crawl in and then feed on the media. Adult flies also will feed on them. So Drosophila has different stages in their life cycle. So you have the embryonic stage, and then there's a larval stage, pupal stage, and the adult stage. Right? So except the pupal stage and the embryonic stage, the other two stages require them to feed regularly. So they feed on the food which is there inside. Right. And we have like different fly stations which you see here, and we also have a specialized microscope which is there inside. Sohela is using that. Hi Sohela. So here we try to express various transgenes which are fluorescent in nature. So we can express GFP, green fluorescent protein, or red fluorescent protein, to mark flies, certain
Right.
flies, or mark certain tissues of the fly, right? And you should be able to distinguish them from the flies which don't express them.
Right.
So in the same population, you may have one set of flies which will express them, one set of flies which may not express them. So that distinction, sorting them out happens here.
Right.
This is a GFP fluorescence microscope,
Right.
stereo microscope again, and we have like a camera attached to it. You can see them, see things. If you need to record something, you can do that
Right.
using the camera setting here. And Sohaila is a PhD student in the lab, so Sohaila, you can talk a bit to
Hi,
Into about
I'm
your work.
Sohaila, and I basically work on understanding how oxygen and nutrition sensing together, they kind of talk with each other, so the crosstalks and how that regulates the metabolism and growth in Drosophila.
Great.
Thank you, Sohaila. Okay, good. Sure. And these are other students in the lab, Ashwati, Nazim, and Anandita. They're all busy working, I guess, but you can say hello and maybe say a sentence about what you're doing, right? A line about what you're doing.
We're collecting larvae right now.
You're collecting larvae right now. Okay, good. So you're also collecting larvae?
Yeah.
Okay, good. All of you are collecting larvae. Okay.
We're basically looking at what genes are involved in growth, development, and metabolism, and how we could study that in the lab using the fly system.
Right.
Good.
I basically work on these small molecules called microRNAs and how they regulate aging.
Good. Nazim?
Yeah, I'm also working on microorganisms, but specifically on developmental stages, how they control each development stages and buffer the development mechanisms.
Right.
Good. Thank you. Thank you, guys. Yeah. Okay, good.
Okay.
You go.
Yeah.
Thank you. Done. So this is a very important space for us. This is where all the growth chambers are kept,
Right.
and the flies are kept inside these growth chambers. So they all have like specific controlled conditions, environmental conditions which are highly specific.
Okay.
So the flies are most comfortable at 25 degrees. That's a normal temperature, and the humidity around like 70 to 80% humidity, relative humidity. So, but for the experimental purpose, sometimes the flies have to be kept at 18 degrees to slow them down in a way that you don't have to take care of them very often. So, storage of the flies mainly happen at 18 degrees,
Right.
and experiments happen usually at 25 degrees, and in some cases it may also happen at 29 degrees. So, we have 3 different temperatures here: 18, 25, and 29. Depending on your requirement, you keep the flies there. So, we can open one of the incubators and see the flies inside. Okay, this is how the incubator looks like,
Right.
and these are like vials which are kept. Again, I showed you fly in the fly room. I showed you the flies, right? So
Yeah.
this is like, these are the same vials
Right.
which are kept here, and you have to keep monitoring them, change them like regularly, and take care of them on a regular basis. So we have these fans which circulate the temperature and the humidity throughout the chamber, and it's all well monitored. So we can change conditions. And also the lights are also controlled by a timer here. So we have a day and night, 12-hour day, 12-hour night cycle which is running. And if you need to have like make any changes in terms of light, if you're specifically working on some certain conditions, if you want to change that, that's also possible. We also have other smaller incubators elsewhere where we can closely like change things and monitor them more carefully. So that's in Dr. Nisha Kanan's lab. Whenever we need them, we go there and use them. Right? And here is a setup which so they need to be fed. You sow the fly food in the vial, right?
Right.
So they need to be fed like regularly, which requires the fly food to be made and cooked. So that's a machine which helps us in making the fly food. That also helps us in delivering the food to these vials. So this machine helps us in automatically pouring them into these vials. So the distribution of the food happens. That is, food is made there, and distribution is happening using this mechanism.
That's
So
interesting.
yeah, good. So that's basically what we have. It's an air-conditioned room, and we also have dehumidifiers here
Right.
to keep the humidity like constant all the time. Right. We need to keep them like very stable That's very important for our experiments because we are looking at changes in the environment, right?
Right.
So we don't want like unwanted changes to happen and affect them. So that changes we would like to manipulate them and see what happens. We won't let the environment touch them directly. So these are very important for us.
We can start.
Cool, sure. These are also bigger ones, similar incubators.
It's the old ones. So Jishy, a lot of students are opting to go for PhD positions and other project positions.
Correct.
So, in your opinion, what are some of the qualities or attributes that a student should have to become a successful researcher?
So I think the key thing is the student should have a good grasp of the basics of the field, whatever research topic they are interested in. So they should have done their masters project in a thorough way, so that they are aware of the basics of various things in the field. And but that is not the only thing, because research requires special capabilities and abilities, right? So what is really helpful is their ability to read and understand things which are like quite deep, right? So they should be able to read papers and understand what's going on in a research field. They should also be able to think or also figure out what is yet to be known, what is not understood in a field. And then they should be able to propose some novel ideas, novel research options for the field to go ahead. So normally I try to test by asking like few questions that way, right? Whether this person can think, whether this person can think of a new experimental option and then try to implement that. So they should have the ability to think independently and challenge a problem and try to tackle that. That's one thing. Another important point is this person should be able to network and collaborate with each other, and should show some motivation and interest in the proceedings of what would be going on in the lab. So by talking to them, we can again figure out whether this person is actually serious or motivated or not. So once you find a person who is motivated, then things are very easy, right? Because you don't need to micromanage them. They kind of take care of their own projects. I prefer not to micromanage people. So I've been trained that way. So I prefer not to micromanage people. And so those who are like really motivated and have the self-drive will do on their own. And one need to monitor them once in a while, and then that's it. So I think these are the key aspects that I would look at. And I think I've been quite fortunate that I've had such students so far. And people actually ask me, what is your formula of getting good students? I don't have a good answer, but this is what has helped me so far. And this is what has been working with me so far. So that would be my answer to that, yeah. Cool.
Being in the field of academia comes with a lot of stress and pressure. And so how do you manage a healthy, friendly environment in the lab?
Yeah, that's a very important but very difficult question to answer, actually. One way to tackle stress is to keep things simple. That's what I believe. Maybe there are other ways to do that. So keeping things simple doesn't mean that you have to reduce your compromise or anything like what you're doing. You don't have to compromise the quality of your research. But if you keep your expectations closer to reality, then I think it's easier to achieve that. And then I'm sure, because we are all intellectual people, we are always trying to do better. When you reach there, I think we'll always try to do things better. So I think trying to achieve something, you're there and trying to achieve something which is far away is much more challenging, much more difficult. One has to do that also. But I think slowly you can make steps towards that and then try to reach there. I think that's probably the best option. But then I think I'm saying all that, but when it comes to reality, it may not be the same. There are moments where I also cannot withstand that And I think the best option then is to always do on a daily basis, do things which helps you to tackle the stress, not just when the stress comes, but you have to always do like things on a daily basis. Prepare yourself, your mind and your body in a way that you can handle that stress when it really comes. So it's thinking that you'll handle stress only when it comes, I think is probably not going to work. So I think this is my approach so far. And then there are various ways in which I do things on a daily basis. I think diet is one thing which I try to take care of, regular exercises, and then socializing and spending time with others. These are the aspects which I try to take care of.
Jishy, let's get into some of the broader questions. So, this is an age where people are moving away from fundamental questions, and with funding agencies always asking for immediate translational benefits, it's getting even more harder to people to get into this field.
Sure.
So, how would you claim that the relevance of basic research is not yet?
Yeah. It's a question of profound interest. It's also very complex to answer in such a short meeting. But I feel that both translational research and the basic research should go hand in hand, right? So a lot of earlier work was basic research. Now I think the relevance is more for the translational, emphasis is more on the translational research. But if you think about like, we can think of our immediate translation, this thing would be human disease conditions, right? And if you think of like various human diseases, like for example, there is this craniofacial defect which can happen. It's called the PRS, the Pierre-Robin syndrome. That happens mainly due to the problems in the formation of the facial skeleton and the muscles in the face. And this can be like, in some people it can have deformities, in some people it can lead to death. So the basic reason for this happening is there are a specific set of cells which migrate when the neural tube is being formed. It's called the neural crest cells. They will migrate from a region where the neural tube is being formed. These are a different set of cells. They are multipotent. Then they migrate to different parts of the body and help in formation of the skeletal system, the muscle system in different regions, though they originate from the neural tube in a focused area. So the understanding of the neural crest cells and all the research which went behind the neural crest cell developing and migration and differentiating into other tissues happened in model organisms and happened at a very basic level. So this was the basic research which led to our better understanding of a disease condition. For example, a lot of cancers are caused due to defects in the wind signaling pathway. Wind signaling is nothing but wingless in Drosophila, which was identified as a wingless fly to begin with. So this was again a very interesting observation which curious people have made, has led to a better understanding of cancer at human health and stuff like that. So that's what I'm saying. So if you try to focus on only translation, that won't work. And so this is biology which we are doing, right? So I'm staying away from the funding agencies. Like my own opinion would be like, we are doing biology. It doesn't have to be human alone. So we have to address questions which are relevant to the entire biome, right? So all different organisms, which includes humans as well. So by nature, I'm inquisitive. I'm interested in understanding how an organism function and how they survive in various conditions. But a lot of these understandings which we gain by studying at a basic level can easily be translated. So you just need to tell the funding agency that we do basic stuff, but we will give you information which can be translated by other people. So I cannot do translation and basic at the same time. That would make it very difficult for me to handle.
We've sort of come to the end of the interview. Just one final question.
Sure.
If you could change one thing in the way that science or research is carried out, either in this institute or in India, what would that be?
So if I compare myself when I was a PhD student in NCBS, and the situation what we have now, I feel that things have become much more easy in India. So the kind of ease to do research in India has improved, and that is a good sign. But there are still things which are challenging. So every institute has its own set of challenges. So when I talk to others, when I tell about my problems, they have their own set of problems, and that's universal, and that is there. But there are specific challenges which we have here. The main thing is like our remoteness. So we are like really far from a decent airport where you can receive things. Customs has some restrictions here. Some things have to come to Bangalore or Hyderabad or even Bombay, and then you're waiting for that transit for those things to reach here. So for example, we work a lot with drosophila, right? And we need to get live organisms here. So after they reach the Indian soil, it takes an extra week for them to reach here, which are things which we should try to avoid. There should be some ease of doing research that way. The time which is taken for transit from the US to India is like 1 day, but from one city in India to another city in India takes 7 days, which cannot be justified. So I think those are the things which needs to be changed here.
So yeah, it was great sitting with you and talking about.
I really enjoyed it. It was really nice. Yeah.
And it's been a great experience working in your lab also for the past one and a half years.
Nice to know that.
Yeah.
Good.
So yeah, we've come to.
And thanks for the opportunity as well.
Yes,
right?
yes.
Yeah,
I mean, thanks for accepting our offer. So yeah, that's
Cool.
it's great.
Cool.
And thank you so much.
Thank you so much.
Yeah.
Thank you. Cool. Sure. Bye.